Okay, so macromolecules, these are why they're important because they store energy. Now, I'm using these values because it's consistent from what I hear with the food science and nutrition classes, so I'll try to keep it consistent between that, even though we're different departments. I think this is good because there are more precise values. I think sometimes going to the hundredth, and even then, it's kind of like a debate. There's always saying that, like, okay, exactly how much energy do you get per gram of a macronutrient? So carbohydrates, 4 kcals per gram, and then fats and lipids, 9 kcals per gram, proteins, 4 kcals per gram. So this is one potential math question, right? So this is, I'm going to keep it simple and say, like, okay, if I ask you, okay, how many calories and 20 grams of carbohydrates, you would say 4 times 20 is 80. So things like that. This is also like the basis behind things in your food labels as well. Nucleic acids are negligible because, again, unless you're absolutely starving and don't have any of those other macros in your body doesn't really metabolize nucleic acids into energy. And then I'm putting ethyl alcohol as well, and ethyl alcohol is drinking alcohol. So it does have calories, it does have energy per gram. And these values are for PHYL 1 for 2. I think these are supposed to be consistent with fission, but for other classes, use their values as well. Now, why did I put this in this order? Well, this is kind of like a rough order. Your body is converting all of these to energy at one point, but the thing is that your body is going to try to use up carbohydrates first and then fats and lipids. That's again why it's so hard to burn off that fat and triglycerides in the gym, right? I wish we could burn that off first. And then proteins, yeah, you want to conserve those because why? Well, what's made out of proteins? Those functional parts of your cells, or if you're one of the people who like to do bodybuilding, like our anabolic examples earlier, do you want to break down that first and then lose all that hard gain muscle? And what important muscle in the center of your thorax is always beating and made of a lot of protein? Yeah, so you want to kind of spare protein. You don't want to really use it if you don't have to. So this is kind of like a rough scale on which macromolecules your body prefers burning first. All right, so then here's our chart again, but this is what we're going to try to cover today in the time we have left. So we're going to talk about like how do we metabolize glucose? So how do we, or not just glucose, but carbohydrates in general. So how do we go from carbs to energy? Now this might seem, well, one thing is you have to break it down into simple sugars. The classic one we saw in that previous metabolic equation with cellular respiration is glucose. Now glucose isn't the only type of carbohydrate, but it best exemplifies that basic pathway of going from a carbohydrate and converting it into energy. So glucose in its ring structure looks like this. And again, this is whenever you see this kind of structure, it's an organic chemistry representation of a chemical. The thing is, or let's rewind that a bit actually. So glucose is less energy than fats because again, 4 kcal per gram compared to 9 kcal per gram for fats and lipids. But the good thing about glucose is it has a pretty direct and efficient way of converting it into ATP. So your cells, they love glucose. They have an easy time converting it into energy. Now, whenever you see this kind of ring structure or this kind of chemical structure, when it doesn't explicitly say what an atom is, it's typically carbon. So this is what we have with glucose. It's in this ring structure, or actually regardless of structure, it has 6 carbon atoms. Now this is glucose over here, and in this equation, remember, we combine glucose and oxygen and make energy out of it. Now what we have here is that how do you get from glucose to ATP? Well, the interesting thing is that it's not like a simple, even though this describes the overall reaction, it's not a single step. Unfortunately, there are many, many steps between that and actually generating energy. And that's the tough thing about this chapter in the Martini and the OpenStax version. They're basically taking an entire 400 level course, like there's biochemistry 441, and then they're trying to condense it all into one chapter and make it introductory. So if it is a tough, challenging chapter, it's because they're trying to do that. But I pretty much keep it limited to what I cover in the lectures. So this is my big study tip for metabolism. You're free to study the rest of the stuff, but keep it limited to what I cover. All right, so then if you've taken biology or like AP biology or intro to biology, you may have heard this term, glycolysis. So glyco is, again, that root word that means sugar. Lysis means rupture or breaking apart. So what you're doing with glycolysis is breaking apart a sugar and also into a form that's more easily metabolized and converted into energy. So do I expect you to know all of these? No, not necessarily, because again, all these like intermediate, those other dots that connect, like, so what I want you to go is basically know that you go from glucose and then you end up with this. Now these three, all these into the middle, that's something that you probably will have to learn for biochemistry 441 if you're a bio major. But again, and maybe if you're going taking the MCATs or advanced class or tests for the for medical or PA or dental school. But again, for this class, I'm more concerned about starting products or starting reactants and end products. Okay, so again, the glucose has 6 carbon atoms. So again, this is our ring structure right here. But this is another way of representing glucose in its linear form, and this will be easier to visualize. So again, 6 carbon atoms in glucose. Definitely should know that. And the ring structure is just linking these two carbons together. All right, so then with our 6 carbon glucose, what happens to it during glycolysis? Now again, I don't want you to memorize these steps, or you don't need to memorize these steps for this class. You might have to do that for biochemistry. But what happens is that ATP is actually used, and the energy from ATP is used to add phosphate to glucose in the very first steps of that metabolic pathway. And the interesting thing is that once you have this these phosphates added to the structure of glucose, it makes the chemical kind of unstable. And it's so unstable that it's easy to break apart this chemical structure. And that's exactly what happens. So glucose now breaks apart into 2 different molecules. Now these now you have like glucose snapping in half, and now you have 3 carbon fragments. And they undergo their own metabolic changes, and this is what happens. So I'm just demonstrating it here. And they do a little shuffling. So this is pretty much what happens during glycolysis. You're taking chemicals and carbon and shuffling around all these oxygen and other chemical groups attached to these carbons. And then at this point, notice that these 3 carbon fragments, after all those different steps in metabolism, they're now identical. And the point where they become identical is called glyceraldehyde 3-phosphate, or G3P. Again, I italicize this because this is more like FYI. I'm not really important at this introductory level, but this is often like something I see on like practice tests for qualifying things like MCATs and NCLEX as well. So G3P is often like one of those the questions they love to test on. It might be useful. All right, so then what's happening? More chemical changes. These 2 molecules are now identical, 3 carbons each. And then eventually this is how you go from 1 glucose molecule and making 3 pyruvate, or 3 carbon or rewind. Okay, so now you have 2 pyruvate molecules, but with 3 carbons each. So yeah, this is the 3 carbon fragments, and this is how you go from glucose to pyruvate. It's not directly, it has to go through all these little different steps as well. Yeah, so the main thing is that you take a big molecule with 6 carbons and make it into 2 identical molecules of pyruvate. Why is it so complicated and seemingly inefficient? It's like 3 steps forward, 2 steps back. Well, another thing is that it also depends on energetics as well. The thing is that you have to get these molecules identical somehow, and at the same time is like I probably would have to research this myself because I don't know the exact energy values for all of these reactions. But sometimes it's easier to shuffle a chemical from one at a chemical group to one carbon versus another carbon on the same molecule. It all deals with all this complicated chemistry and biochemistry. So yeah, this is why sometimes it's not direct because you have to, just like how, why do you have to add those phosphates? Well, it makes the whole molecule unstable, but it did require energy to actually generate that. This is why glycolysis consumes energy. It actually is, in the first two steps, it consumes ATP, but it's trying to break this apart. Yeah, so it's trying to do a path of least resistance, but with many, many, many different steps. That's very observant. Yeah, so the cool thing is that it's not a net. So when, again, net gain means that the overall result. So the thing is that you do have a net gain of 2 ATP. And the cool thing is that when it does go from here to here, you do regenerate some ATP, but you also need some investment to actually get that 2 ATP. All right, so then the glycolysis occurs in the cytosol, not the mitochondria. So this is this is what I definitely want you to know. It takes 6 carbons, breaks it in 2, shuffles it around with metabolism. And again, these steps I don't expect you to know at this point. And then it makes 2 identical pyruvate molecules, and these have 3 carbons each. All right, so then glycolysis ATP. So again, you need to do that initial investment because again, remember that beginning steps, they try to make the whole glucose molecule unstable, so it uses up those 2 ATP, but does generate 4 ATP as it starts to break apart bonds and shuffle around the chemical groups surrounding those carbons. So you do have an overall net gain of 2 ATP. But the thing is that it also generates not just any ATP, but also a special chemical called NADH.